Hey, yo, is this thing on? Where's the freaking audio? There it is. Hey, yo, it's Rutherford here. We're going to do Unit 3, AP College Board, multiple choice questions. Oh, my class. Okay, so move my freaking face up here. Right. So Unit 3 is our cell respiration, photosynthesis, energetics unit. And okay. Ooh, alcohol dehydrogenase. Never heard of that, right? But we do know what dehydration synthesis is, and ACE is an enzyme. Oh, it tells you it's an enzyme. See, that's the thing about the AP test is like, you got to have a certain amount of background knowledge here, but then oftentimes the questions, they will like tell you stuff. So don't go in thinking this is just a blank piece of paper and you have to have everything held into your mind. You have to be able to solve problems, figure things out. And, sometimes it's rarely just pure memorization. I mean, there's a few of those questions we've gone over, but that's not the main. Okay. So, this one, I do feel like reading the actual question first. I don't know. Enzyme that aids in the decomposition. Not dehydration synthesis, but it's the decomposition of ethyl alcohol into non-toxic substances. This could be happening in the smooth endoplasmic reticulum. Methyl alcohol acts as a competitive inhibitor to ethyl alcohol by competing for the same active site in ADH. Okay, so this is our concept in an enzyme, is that it has active sites and it has inhibitors, activators. It has like its designated binding molecule, which is called a ligand. So yeah, basically you either have methyl alcohol or ethyl alcohol. The same concept is with carbon dioxide and oxygen. So when you have carbon dioxide and oxygen, they take the same spot on the hemoglobin molecule of your erythrocyte red blood cell. And the hemoglobin can hold 4 at a time, each little hemoglobin protein. And so if oxygen's there, then it's full. If CO2 is there, then it's full. You can either have 2 oxygens, 2 CO2s. You could have 1 oxygen, 3 CO2s. You could have 4 oxygen and, whatever. But it looks like this is the same kind of concept here with this alcohol dehydrogenase. So the question, when attached to ADH, the methyl alcohol is converted to formaldehyde, which is toxic in the body, okay? So we do want something to break down ethyl alcohol into non-toxic substances. However, methyl alcohol can sometimes block that binding site where the ADH cannot be. Yeah, if methyl alcohol is there, basically ADH, and this is something we could draw out. Like I could just have this on my piece of paper. It's always ready. I go like ADH, and then I could draw plus methyl alcohol. Okay, you could write meth or Ma equals toxic. Okay, and then you could write ADH plus ethyl alcohol, EA, equals non-toxic. Okay, so ethyl alcohol good and methyl alcohol bad. So that's something that's something I could write down, and I can explain that to you in like about 25 seconds. And that's something that you could easily write down, and it might take 45 seconds or something, but then it just allows you to move through the question so much easier. Okay. A, which of the following statements best predicts the effect of increasing ethyl alcohol? So if we know we increase ethyl alcohol, then non-toxic, okay? But we keep methyl alcohol the same. There'll be an increase of formaldehyde, no, okay? Methyl and ethyl are competing. Ma and Ea, the ethyl alcohol and Ma, they're competing. So A is a no. B, competitive inhibition will be terminated because ethyl alcohol will bind to methyl alcohol, no. That wasn't part of it at all, okay? Ethyl alcohol is binding to ADH. Methyl alcohol is binding to ADH. They're both ligands, okay? And they're tiny little things. You want to think about tiny little things bonding to a protein when we're thinking about these concepts. And C, peptide bonds in the active site of the enzymes will be denatured, inhibiting the enzyme. No, we're increasing ethyl alcohol, and that is decomposed in non-toxic. So I got to go with D just by process of elimination, but competitive inhibition will decrease because of the proportion of active sites occupied by substrates. Look at all these fancy words, okay? We just got to know, I say ligand, they say substrate, same thing, okay? The active site is the part on the enzyme where it binds, and they're competing. Ethyl alcohol and methyl alcohol are both competing to bind with ADH. There's like a little competition there, okay? This is a classic concept. This is not something to just memorize for, like I said, this is oxygen and CO2. It's not something just to memorize for ADH. This is how many, many things work in the whole body, right? You got things, small things competing to bind in a large thing. The large thing is always an enzyme, okay? So if you increase one, then it's just going to win. It's going to bond more. There's just a lot more of it there, okay? Next, atmospheric carbon dioxide. You see how they all start with the same thing? And they say produced byproduct typically enters plant leaves, raw material for photosynthesis, okay? Yeah. We got to go to the researcher claim. So this is a researcher claim, a justify kind of a thing, or best supports. We'll call this the supporting actress question or something like that. We're not trying to find for questions like this, we are not trying to find the answer that is correct or true. We're trying to find the answer that supports the researcher's claim. So you have to be oriented on the researcher claim. Increased atmospheric carbon dioxide levels will increase growth rates in plants, okay? So A, atmospheric carbon dioxide is produced in the burning of fossil fuels. No, that has nothing to do with plants, okay? Nothing to do with plants. B, atmospheric carbon dioxide is a byproduct of cellular respiration. Again, that's not talking about plants. That's talking about combustion or animals breathing out CO2. We want to be talking about the increased growth rate in plants. C, atmospheric carbon dioxide typically enters plants through the stomata, which plants rely on for regulating gas exchange. Again, that's a true statement, but it has nothing to do with the researcher's claim. So I'm really liking D. Carbon dioxide is the raw material for photosynthesis, straight up, okay? So if you increase it, then which plants rely on for producing, yeah. So it's the raw material. It's the thing needed for photosynthesis. So that would support the claim that the researcher says more of it means more growth. I got to go D. Hell yeah. Okay, another researcher claim. Researcher claim, different metabolic pathways allow bacteria to use different molecules. Okay, so we got different metabolic pathways, different molecules as sources of matter and energy. Okay, so we're going to look for some sort of comparison. A says rhizobia bacteria form close association with the roots of bean plants. No, we're not talking about two different bacteria. Can't be right. B, E. coli bacteria, media containing, again, yeah, that's a bacteria, but I'm going to look for one. C, the antibiotic rifampicin inhibits the growth of some bacterial strains, but not others. Not a comparison of different molecules, different bacteria, but D, some viruses that infect bacteria reproduce by either the lysogenic or lytic cycle. Well, D is dead wrong. That has nothing to do with bacteria making energy. That's a virus. So now this is a cool example of like, I go through them and nothing sounds right to begin with. I know that D is trash. Okay. Let's go back to C. The antibiotic inhibits the growth of some bacterial strains, but not others. That is a comparison. We are talking about two different bacteria. Inhibiting growth, you are kind of like, well, that doesn't talk about matter and energy, but energy is required for growth. And C is comparing different bacterial strains. Okay. So I do like C, but let's go B. E. coli reproduce in liquid media containing glucose or galactose. Yeah, but it doesn't say anything about different bacteria. So we do have different molecules. Different metabolic pathways allow bacteria to use different. Oh, I do like B. I all of a sudden like B. Okay. So let's double check A. The bacteria form close association with the roots. Again, that doesn't have anything to do. It's got to be B. Okay. Now that's the double check situation. And I'm on B there. That's exactly what it says. So I was reading it a little too quick and just kind of zoomed ahead, but that's a evidence of it's fine to read questions quick as long as, because then you can kind of weed out like the ridiculous ones. And then for me, I just narrowed it down to B and C. And then I could go in and really zoom in and remember, see, I went back to the question and I was like, okay, so read different bacteria. No, it was different molecules for energy. And that's what really settled me on that E. coli one. Okay, now another researcher claim. You got to be kidding me. Okay, we're getting so good at these. The only portion of the light energy captured is available, that only a portion. Okay, which is true, right? We got Roy G Biv, and the plant reflects the G, which means it's not absorbing G. It is absorbing Roy and Biv. Okay, so we got to justify that. Light capturing pigment in green absorb the red, blue, and violet lights. I mean, honestly, if I'm going fast, that's A. I don't need to. Oh, okay. So I screwed up. Not a big deal. But I think that was a good choice. C is right, though, because it says as light energy is converted to chemical energy, some of the energy is lost as heat. Oh, interesting. Okay. Researchers claim that a portion of light captured by green plants is available for growth and repair. You know, that's really interesting that A is not the correct answer. That's pretty tough. We can look at B and D here. The energy of photon light is proportional to its B is just a stupid definition. And D says captured energy is stored in the molecular bonds of organic molecules. Again, D is just a simple definition too. B and D are pretty clear. Like, yeah, the researcher's not talking anything about light, really. I mean, light and growth and repair, energy capture, light capture. That's not, I mean, I guess it's talking about that stuff, but it's not specific enough. But I guess looking at C again, as light energy is converted to chemical, some of the energy is lost as heat. Okay. Yeah, only a portion of the light energy is available for growth and repair. I do think C is slightly better than A. And this is where, you know, you'd be like, well, they both are correct. Yeah, but they get you with that. Which of the following observations best helps support the researcher's claims? Best. A is good. C is better because it's explaining that only a portion, right? So we're looking at that word a portion and we're saying some of the energy. A portion of the light energy, some of the energy. That phrase is literally identical in the researcher's claim and the proof in C. 10. Gosh, there's no diagrams on this. What the heck? There's no graphs. There's no diagrams. It's boring. Research designs an experiment. Okay. Which of the following must remain the same for all trials of the experiment? Okay. So this is testing your ability to design a good experiment that is, has similar kind of setup and controls. The experiment to investigate the effect of environmental temperature on the function of an enzyme. So I can imagine they're going to, the independent variable is temperature. They're going to change that. And then they got to see how the enzyme performs. The researcher will add the enzyme and its substrate to an aqueous buffer solution. And remember, enzyme, protein, substrate, small little thing that binds there, a ligand, okay? To an aqueous buffer solution. I mean, it's just fancy words. It's just in liquid. And then measure the amount of product formed over 20 minutes. So obviously they all have to have 20 minutes. Okay. That has to be a fair race. Each, they're going to be changing the temperatures to see, but everybody gets 20 minutes. Everybody gets the enzyme and everybody gets the same amount of the enzyme, the same amount of the substrate. All of that has to be exactly the same. And the only thing we're changing, the independent variable, is temperature. So, which must remain the same for all trials of the experiment. These are called controls. The initial concentration of the substrate, hell yeah. The final concentration of the product, no, that's obviously going to change, okay? That's who wins the race. C, it's got to be A. Three-dimensional structure of the enzyme, yeah, I mean, that has to stay the same. No, because remember that whenever anything bonds to an enzyme, it will change shape. But also C is like, I mean, you got to use the same enzyme, but A is just perfect, right? Like you have to have, everybody has to be starting with the same materials. And the only thing we're changing is temperature. D is the dependent variable. That's like, or D is the independent variable. That's exactly what we're changing. B is the dependent variable. That's what's going to change as a result. So we change D, B changes as a result. C is weird. And A is very clearly the control in this experiment. Shazam. 12. Oh, finally, a little picture showing an experiment. Okay, great. So it's an experiment question. Let's just go straight to the question. Which of the following would be the most appropriate control? Again, a control. Before they said stay the same, now they're saying control. They're just testing your knowledge that those are the exact same things. So what do we have to keep exactly the same here? A test tube containing no camera film, only a piece of exposed camera film, a test tube containing a piece of exposed camera film submerged in water. So we just, gosh, they're just keep getting longer and longer. Remember what I said before, where it's like the longer one might be the correct answer? I don't know if that's the case here, but let's just kind of see. Based on this experiment, if I don't want to take the time to read this, I'm like, the diagram for experimental setup does have a piece of camera film in an enzyme solution under water that is the temperature is being changed. And then you've got the test tube. So I don't know exactly what they're changing in this, but we want to think about the thing that should not change. So I guess for that, I'm going to look up here and see, let's look for the things that's changing, and then we'll know what should not change. So the independent variables, what we change, dependent variables, what changes as a result. And then the control is like, listen, we got to, be doing everything the same for all the experiments. Gelatin, collagen, here they're giving you information here. You don't have to have those memorized. The enzyme to digest gelatin, camera film has gelatin. Okay, great. And I would be underlining little things here. Students incubated camera, each containing one of 3 different enzyme solutions. There it is. Okay. 3 different enzyme solutions. Right there, I'm writing IV. That's our independent variable. And then they record the time it took for the enzymes to digest the gelatin in the test tube. Okay. So the amount of time is the dependent variable. Okay. And the gelatin is going to be digested here. So what needs to stay the same? The freaking temperature. But that's not an option here. So let's try the control in this experiment would be the one that we just want to see. Okay. The control is a good example of they want to test it and just see if, let's imagine, okay, we're putting 3 different enzyme solutions. What if we just put no enzyme solution? What if we put the exposed camera film in just water? Okay. And see like, okay, because maybe the camera film was just digesting in water. It has nothing to do with the enzyme. So I know that that's going to be the right test here. And a test tube containing a piece of, so I like C here. All 3 enzyme solutions, that's like no, because then you won't know which one is actually doing the work. B, a test tube containing only a piece of exposed camera film. I guess you could kind of see what it's like in air, but that's not really the same as the enzyme solution here. And then A is like, well, duh, if there's no camera film and the test tube's just sitting there, like you're not going to see anything, but we want to see what would happen if the camera film is just in water. Okay. That's how we're going to see here. So I know we've got C. Thank you. Okay. On to 14. Finally, a freaking ATP question. Okay. Blah, 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 blah. Which of the following statements provides evidence to support the claim that no ATP will be synthesized in the absence of a proton gradient? Okay. So this is kind of like a malfunction scenario here. So if you don't have a proton gradient, you will, this is just classic memorization. You will, if there's no proton gradient, you are not, building up and going from high to low through the ATP synthase, and you're not going to make ATP. Okay. This is just, I'm not even reading the first part because it's pretty clear. Blocking electron flow from one carrier to the next in the electron blocks formation of a proton gradient. Yeah, that's true. Let's try B. They're talking about moving from, in chloroplasts here, they're talking about moving from photosystem 2 to photosystem 1. But B, increasing the proton concentration difference across is not associated with increase in the rate of No. B is like, yeah, if you got more protons concentrated outside of the thylakoid there, they're going to want to flow back in. And yeah, so B is exactly how you make ATP. Here they're saying, which provides evidence to support the claim that no ATP will be in the absence. So B is kind of like a more ATP with more of a proton gradient. So B is kind of saying the opposite of what the researchers claim is, which could prove, if you can prove that the opposite is true, then maybe, but I don't love it. C and D are probably going to be looking like the ones we want. Okay. Again, no ATP will be synthesized. It says it right there. Okay. And then it says it in C and D. So I'm kind of like leaning towards those two if I'm moving fast. Channel proteins that allow the free pass of protons are inserted into the thylakoid. Oh, yeah, I like that. Okay. So if you have channel proteins, then the protons can just balance out to equilibrium and they won't be flowing through the ATP synthase. Okay. So I am really liking C. That would definitely be a no ATP situation. And then no ATP is synthesized while the Calvin cycle is synthesizing carbs using ATP. No, that has nothing to do with the proton gradient. D is just like, has nothing to do with the proton gradient. I'm feeling pretty confident with C here. Okay. Yeah. So notice that B, where I had like, okay, that's the opposite, but proving that someone with shoes can run doesn't really mean that someone without shoes can't run. So that's why we don't like B. Okay. Great. All right. Hopefully you can't hear the airplane outside here. I'm stoked. This is a great little session here. I'm at my standing desk, standing up on this. I just like put my laptop on a freaking chair or like something so I can stand here and move around. That helps the brain work really well. And I did a quick little workout right before this, just 10 minutes. And, that gets the brain juices flowing, gets you feeling a little more positive and much more productive and able to think. So, finding the things that work for you to get your brain active. Oh yeah. Oh yeah. So don't stand up all day, but standing up when you need to use your brain for short bursts of time, like 30 minutes or something. Oh yeah. All right. Which of the following best supports the claim? Another one. Okay, that species is better adapted to environmental changes than other photosynthetic species are. Okay, this species. Oh, in addition to the pigments, a certain photosynthetic species contain, it's just like they're imagining a freaking species that has 2 additional pigment types. Now, in normal plants and stuff, we've got the P680 and the P700. I think that's what it is in photosystem 1 and 2. Now they're just saying there's more. So I'm thinking like, okay, well, if we have pigments that absorb different wavelengths, and then we have 2 additional ones, we'll be able to absorb more light, right? Okay. So which of the following best? Okay, we got to underline the word best because we're going to find some correct answers here. We want to find the best one here. A, the increased pigment concentration better facilitates energy production within the cell. It's not the concentration of the pigment. There's just different pigments. So I don't love A to begin with. B, the pigment combination. There we go. Different pigments allows the organism to absorb heat as well as light. Heat, they are testing your physics knowledge that heat is on the energy spectrum, where the light spectrum goes Roy G. Biv. When you start getting lower than Roy, you get into the infrared, and then you get into just like heat energy, because it's all just energy. Some energy is light energy plants can absorb and our eyes can absorb, and there's heat energy too. Like snakes can sense heat and see with heat. It's just a different version of energy there. So I like B. Let's try C. Additional pigments allow the species to outcompete other species for wavelengths of light commonly used in photosynthesis. I don't think we're outcompeting. There's plenty of freaking sunlight. That's ridiculous. Okay, D. The additional pigments allow the species containing them to harvest energy from wavelengths of light. Oh, D's really strong. D's really strong. I like B, but D seems very strong here. Yep. They're just literally in this. This is a type of question. They straight up rephrase the question as the answer. We had another one of those earlier here. They literally took the question, they rephrased it in the answer. A lot of this stuff, this test taking stuff, this is not about your biology knowledge. This is not about memorization and stuff. It's your ability to just move big vocab words around and just kind of understand like the basic structure of this question here. That's why I'm spending so much time on that. It's like, I can teach you some biology here, but really I'm teaching you test taking here. Okay. Now a graph. Oh, we love them. This is the stuff that I love. Now notice, I enjoy the ones that I love, and I still work really hard on the ones that I don't love. Everybody has their specialty question, but you can still get good at all questions. All right. Which of the following actions will provide the most appropriate negative control for the experiment? Oh, they're testing your knowledge of negative control. Honestly, I don't really remember this one. So we'll see if we can kind of figure it out. We kind of know what a control is, something that, with our last one, it was like, oh, we're going to try different enzymes, and then we're also going to try just plain ass water. Then a negative control, something along those lines. So we got to kind of see the setup here. Different pH ranges. I don't have to read anything else. I literally saw the word different pH ranges, honestly, because pH kind of jumped out at me because it's bold. That's the independent variable. Okay. So we want to see that different enzymes exhibit different maximal functions. So the dependent variable is their enzyme activity. Okay. And it looks as though they're comparing these different ones here over different pHs. The researcher cared to test the claim, 3 different enzymes. Okay, the results of the experiment there. So which of the following provides the negative control? Repeating the experiment with denatured enzymes with a 4th enzyme. A doesn't really make sense. That's just not useful here. Repeating the experiment at several different temperatures, that just complicates things. So I really don't think it has anything to do with temperature. Repeating the experiment with denatured enzymes. So denatured, they're testing your vocab knowledge that denatured enzyme is an enzyme that is just a different shape, so it doesn't work. Repeating the experiment using several different methods for measuring pH. Maybe, but again, I don't think that's negative control. C might work there because we're just kind of like, okay, if we break the enzymes, then will they still be active at different pHs? Gosh, a negative control. We're going to have to see their explanation here. Honestly, it's okay to be stumped by a question here, and I think I'm just going to admit defeat. Always take your best guess. And so for me, my best guess, I don't think a 4th enzyme really helps. Negative control is I think it's when it's broken. So I think negative control is just like, let's break everything and see what happens and see. Yep. Okay. So negative control is like, all right, so we got runners in a whole bunch of different types of shoes to see how fast they can run. And let's try runners with no shoes or something like that. Like, let's just break it entirely. Yeah, good. Okay. 2 more. Let's freaking go. Oh man, 20 is okay, fine. Let's go to 22. Hopefully it's oh, good. Okay, so we got some graphness here. A researcher claims that increasing the environmental temperature by 10 degrees C, this is the last one. Notice I didn't jump to the answers. I'm just over here reading the actual question. I get so excited, but I'm going to go down to the question. Which of the following statements best helps justify the inclusion of test tube 5 as a control? So test tube 5 is the control, temperature at 25 with a substrate, enzyme, and pre-treated. So basically with the control here, number 5, they just did everything. Okay. Whereas in number 1, they had no boiling. In number 2, they just had the enzyme. In 3, they just have the substrate. 4, they have no boiling, but they're changing the temperature. And then 5, they're like, all right, let's do everything at 25 degrees. So 5 is just the thing to compare everything else to. It will provide a measurement of product formation in the absence. No, we have substrate. Oh, this is so easy. Provide a measurement of product formation in the presence of denatured enzyme. No, because there's lots of them with enzyme. A lot of them have enzyme. Or denatured enzyme? No, it has nothing to do with denatured enzyme. Everybody gets the enzyme. Nothing in here says denatured. What the hell? No, absolutely not. C, it showed the effect of doubling the amount of substrate. No, it doesn't have extra substrate. It just says yes with all the other ones. So it's got to be D. It will show the effect of increased enzyme activity on the rate of product formation. Okay, so here they are saying increased enzyme I mean, by process of elimination, A, B, and C are trash. But what they're asking for in D is it will show the effect of increased enzyme activity on the rate of product formation. What they are saying is, all right, listen, let's add the substrate and the enzyme and pre-treat it with boiling. But that is denatured. I am now seeing, this is very important when you're looking at data. Always circle the weird thing. Okay. So here, test tube 1, no substrate. Test tube 3, no enzyme. And then over here, no, yes. So what's weird about test tube 5? The only one that was boiled beforehand. The only one. Okay. Lots have enzymes, lots have substrates, lots are at 25 degrees Celsius. The only one that was boiled beforehand. So they are testing your knowledge here to know that if you boil proteins, enzymes, they will unravel. Okay. You won't break their primary structure, but you will break their tertiary 3D structure. They will unravel and be non-functional. That is called denatured proteins. Therefore, B is now officially the right answer. Okay. So again, you notice this, I don't have any pride. I don't have any ego with this question. You know, I ripped through these and I was like, A, B, and C are trash. It's got to be D. If I was moving fast, I would have picked D and I would have gotten it wrong. But you can always stop and reevaluate and be like, wait, there's something I missed. And I'm over here teaching you this, right? And I realized I missed this really big red flag right here. Test tube 5 is the only one that was boiled. Okay. So it's fine to just like sometimes trust your gut, and other times it's fine to just realize, oh, my gut was totally wrong. Like I just missed that. I didn't see it. So I'm liking B on this now. Pretty clear. Shazam. All right. I appreciate you jumping in on this. Enjoy the rest.